All right. So, friends, family, colleagues, mentors, and mentees, and distinguished examiners today, I'm really excited to share with you the results of my PhD and what I've been doing for the last 5 years. I hope you'll enjoy this seminar, and without further ado, I will get started here. I will share with you, oh, something happened. I will share with you a theory of nanoparticle dosing. And to get everything started, I want to start with a little bit of background. This is Sidney Farber. He was a pediatric pathologist back in the 1930s, 1940s at Children's Hospital, Boston. And he's the inventor of modern chemotherapy. Right, so back in the early 1900s, a lot of children with leukemia.
Ben, before we go, can you make it a full screen?
Oh, is it not full screen?
No, it's not full screen.
How about now?
Yes.
Now it is?
Yes. Great.
Okay. So he's an inventor of the modern day chemotherapy back when childhood leukemia was essentially a death sentence, right? The children didn't have very much time to live after they were diagnosed. And so he was a pathologist, and he got to experiment with the samples from the patients. And so he took blood cultures from patients and he put them into petri dishes. And there, he tested various hypotheses, right? He noticed that these cells divided really quickly. And he said, okay, if I can inhibit some sort of DNA replication, then perhaps I could get these cells to go away. And so he tried in a couple of compounds in the petri dish, and it worked, right? These drugs seemed to stop the leukemic cells from dividing. And so he said, you know what? If it works in the dish, maybe it'll work in humans as well. And so in 1947, he started the very first human clinical trial of chemotherapy, right? And he injected his compound into 16 children with acute leukemias. And in 10 of these 16 children, they achieved temporary remission of their disease. And so at a time when this disease was supposed to be fatal, Dr. Farber had invented this new procedure, right, and discovered this new therapy that breathed life into and hope into these patients, their families, and doctors all around the world who were caring for these patients, right? And so we jump to today, and we know that chemotherapy has extended and cured the lives of millions of patients around the world, right? But of course, we all know the side effects of chemotherapy. Right? When a patient receives chemotherapy, their hair will fall out. They'll have nausea, vomiting, diarrhea. They'll be subject to these deadly, life-threatening infections. And it's because of the way that chemotherapy is delivered to people, right? It's injected into the veins, and then from there, it's spread throughout the entire body, and it accesses all the cells in the body, and it essentially prevents any cell that divides rapidly from dividing, right? So hair cells, your gut cells, your white blood cells that are supposed to keep your defenses up, right? All of these cells are hit non-specifically, and they go away, and they cause all of these dangerous side effects. And so pretty much since the beginning of the invention of chemotherapy, scientists have looked for a magic bullet solution. And what is this? What does this mean? It's the idea that there would be some compound out there that would be able to hit the cancer cells specifically without touching any of the healthy cells, right? And so you would be able to crank up that dose, you would be able to kill the cancer cells, and the patient would be fine otherwise, right? And so, this is not such a crazy idea because we've already seen it in other fields, right? We've seen it in the field of microbiology and antimicrobials, right? So when we get sick with an infection and we take penicillin, we don't normally expect to have many side effects because the penicillin affects the bacteria only. And so we've been looking for this in the field of cancer therapy. And, the first inklings of this started to come out in the 80s and the 90s in the form of nanoparticles. And so what are nanoparticles, right? And I just want to share with you a little bit of intuition here. We've got a size scale here, right? On the order of 10 to the negative 1 to 10 to the 8 nanometers, right? We've got tennis balls at 10 to the 8. This is roughly 5 to 10 centimeters. And we can go all the way down to glucose, 10 to the negative 1, where we're on the scale of angstroms, right? And at that scale, we're looking at atoms. Over here, we've got protein, DNA, and viruses, and these are on the order of 1 to 100 nanometers, right? And these are exactly what nanoparticles are, right? They lie on this scale between 1 and 100. And because we can design nanoparticles at this size scale, it allows us to communicate with biology at the fundamental level that biology uses to communicate with itself. And this is what makes nanoparticles so special, right? And nanoparticles come in many different forms, right? We can engineer them to be micelles. We can engineer them to be liposomes or dendrimers. These are kind of like the organic materials that exist already in our body. Or maybe we can engineer them to be gold, quantum dots, or polymers. All of these inorganic materials that don't normally belong in our body, but we use as experimental therapies, right? And it was found that, very interestingly, molecules in this size range tended to accumulate into tumors more specifically than the healthy tissues around the tumors, right? And so this was the beginnings of that first inkling of that magic bullet. For some reason, these nanoparticles were hitting tumors, and potentially we would be able to target tumors very specifically. Now, fast forward today, nanoparticle therapies have been around for 30, 40 years now. And there's still room to improve, right? So our lab performed a study back in 2016, doing a meta-analysis of the field to really ask the question, how well have we been doing as a field over the years, and where are we at currently? And it turns out that even though nanoparticles deliver to tumors specifically, we're still kind of stuck. There's this, the analysis showed that over 10 years of research in the papers that were published, there was a median of 0.7% of the injected dose that made it to the tumor. And so what does that mean? That means that when you inject 1,000 nanoparticles, about 7 of them will make it to the tumor. 993 will be elsewhere in the body. And so even though this is better than other normal tissues, there's still a lot of nanoparticles that don't make it to the tumor, right? And it showed that as a field, over the last 10 years, we've been kind of stagnant, right? If we had been getting better as a materials field, this should have been increasing. So this showed that there was still some barrier that prevented us from improving. And it's interesting because, as a nanoparticle field, we have so many different ways of improving the way that we deliver nanoparticles, right? We can change the material. They can be inorganic or maybe they can be organic, right? So inorganic may be your gold, your organic may be your lipid nanoparticles. We can also change the targeting strategy, right? So we can put targeting ligands on it that specifically recognize cancer cells. We've changed the size, the charge, the shape, and we've even played around with the biology to see if different nanoparticles work for different biological applications. And overwhelmingly, what we see is that there is this 0.7% delivery efficiency across all of these, which suggests that there's still some factor that we don't understand that we need to change to make it to make things even better. So what's the problem? The problem then is the liver, and the liver is the biggest barrier to nanoparticle delivery. And why is that, right? So here I'm showing you an X-ray of a mouse, and this mouse has been injected with nanoparticles, and essentially anywhere where you see a bright signal, that's where the nanoparticles are, right? And so we have red, yellow, white, and what you can appreciate is that this mouse has a flank tumor over here, and you can see that the nanoparticles have accumulated quite specifically inside of this tumor, and there's not that many nanoparticles elsewhere in the healthy tissues of the body, right? Which suggests that the nanoparticles are getting to the tumor. But what you'll appreciate here is that liver is overwhelmingly bright, and it's super, super saturated with a bunch of nanoparticles, and it's taken up a lot, right? And this is one of the biggest problems. The liver, you can imagine, is this, intuitively, it's this blood filtration organ, right? It clears compounds and it clears particles, and so when you inject nanoparticles, they all end up here, and that makes sense, right? So, knowing that this is the problem, I come to the big question that governed the majority of my PhD. And the question is simple, right? How do we overcome the liver, right? And so the thesis, I had 3 goals for this thesis, and they were broken down into these themes, right? First of all, I wanted to understand the system, right? How do we overcome that liver? Well, we need to first understand the interaction and the dynamics and differences between the liver and the tumor. Next, once we understood the system, then we would be able to develop a principle. And this principle would allow us to change the amount of nanoparticles that enter the tumor, right? And finally, once we understood and developed the principle, we could use that principle to invent a solution that increased the anti-tumor efficacy of nanoparticles, right? And so we'll go over these three today, and I'll introduce them to you. But really, the meat of the thesis will lie in development of this principle. And with that, we'll get started. So trying to understand the system, the liver versus the tumor, right? The liver takes up so much more than the tumor. The question is, why? Why is the liver so avid to taking up nanoparticles? And so when I first joined the lab, Dr. Soy and Dr. McFarland were on the verge of figuring out the mechanism of the nanoparticle clearance by the liver. And what they had discovered is that the liver has these unique flow properties, but importantly, that the main cell type to take up nanoparticles in the liver are the Kupffer cells, right? They're the macrophages that just kind of clear and eat everything that passes them. A couple months later, the study led by Dr. Tavares, Dr. Poon, and Dr. Zhang showed us that if we eliminate the Kupffer cell and we remove them from the equation, then the liver accumulation was less and the nanoparticles were able to deliver to tumor better, right? So this cinched that relationship between the liver and the tumor, right? And it told us that, if we removed the contributions of the liver, we would be able to increase the effects of tumor delivery of nanoparticles. And so, knowing this, we wanted to compare the two cell types within the liver and the tumor. In the liver, we know that as the Kupffer cells. Well, what cells take it up in the tumor, right? And so we ran the experiments to do this. And what we did was we injected a mouse with gold nanoparticles, inert, no biological activity. They're just a tracer particle. And then 24 hours later, we took out the livers and the tumors, and we turned them into single cell suspensions, and we analyzed them. We asked, what cells are taking up these nanoparticles? And what we find is that overwhelmingly in the liver, no surprises, it's the macrophages that are taking up the particles, right? But interestingly, we find that in the tumor, it's also the macrophages that are taking up the nanoparticles, right? And so we said, if the macrophages in both organs are taking up the nanoparticles overwhelmingly, then what really is the difference between the macrophages in the liver and the tumor, right? And so we ran a couple of in vitro experiments, and what we found, surprisingly, is that in vitro, the tumor-associated macrophages are more phagocytic than the liver macrophages, right? And so this was a little bit counterintuitive because it didn't explain why the liver took up more as an organ, right? And so we did a few more experiments, and ultimately what we found was that really the macrophage position determines the uptake in these organs. When you look inside of the liver, so this is a transmission electron micrograph, super resolution of the liver. We have a hepatocyte here, hepatocytes. And what we have here is a red blood cell, right? Which indicates then that this structure here is the sinusoid, the blood vessel of the liver. And you'll see over here that we have a cell over here, and this is a Kupffer cell. And it's filled with these black dots, which are the gold nanoparticles that we injected. Now you'll appreciate that these cells are in direct contact with the sinusoid. So any nanoparticle that passes by has to make contact with this Kupffer cell, and that allows this Kupffer cell to take up a lot of nanoparticles. In the tumor, it's a little bit opposite, right? What we see is that we've got a blood vessel here with the red blood cells, but that the macrophage over here actually lies outside of the tumor vessel, outside of the endothelial cell and outside of collagen. And so any nanoparticle that has to make it into the tumor has to first get through the endothelium, get across the collagen to eventually make it into this macrophage, right? And so we said, is this something that is just a correlative visualization of what was going on, or is this causative, right? And so we did a very simple experiment in vitro, and we performed a transwell experiment, right, where we looked at different configurations of these cells. And so we put nanoparticles on one side of this transwell, and then we seeded our macrophages and endothelial cells on opposite sides of the transwell. And so over here, we've got endothelial cells acting as a barrier to the nanoparticles, and the macrophages are indirectly exposed. And we flipped it, and we said, okay, well, sometimes the macrophages can be directly exposed. And that's kind of like the liver configuration, right? And no surprises here. What we found was that if there is the barrier where the endothelial cells are in front and the macrophages are indirectly exposed, they took up fewer nanoparticles than if the macrophages were directly exposed to the nanoparticles, right? And so this told us that the liver has this affinity for nanoparticles simply because the cells are intravascular. They have direct exposure, right? And with this, we were able to theorize something, right? biology, the cover cells in the liver, they are biological entities, right? They have to be finite in their abilities to do certain things. And so we hypothesized that, at some point, if we had too many nanoparticles, at some point, these liver cells would not be able to take up anymore. And, if they are overwhelmed, then they would lose their preferential access. And in that case, then we would get more delivery to the tumor, right? And so this made up our theory of nanoparticle dosing, right? And here we were going to develop a principle to figure out exactly how dose contributed to nanoparticle delivery to the tumor. And so we started this experiment, and we used 3 orders of magnitude, right, for our dose, and we spanned 3 orders of magnitude, and we started by studying the liver using gold nanoparticles. And what we found was exactly as we increased the dose from 50 billion to 50 trillion, we found that the percent of that dose that ended up in the liver started to go down, right? So this suggested that yes, the liver was finite and it was able to be overwhelmed. And then what we showed is that, if the liver itself is taking up less particles and it's clearing less, then there should be more particles in the blood at any given time, right? And this is exactly what we saw. When we started at our lowest dose of 50 billion, after 5 minutes of circulation time, we had already lost 80% of the nanoparticles that we injected, right? They're gone really quick. But if we go to our highest dose, after 5 minutes, we had only lost about 5%. Right, so this is an extremely dose-dependent process. And of course, when nanoparticles remain in the blood, this translates to a higher half-life, right? This is the measurement of how long nanoparticles can circulate. And at our lowest dose, we had, a half-life of 2 minutes. And at our highest dose, we had a half-life of 8 hours. And so you can see here, we didn't do anything to the particles. Everything was the exact same. The only thing that we changed was the dose, right? And this told us that the dose of nanoparticles could really affect how they circulate. And so the pharmacokinetics of nanoparticles is extremely dose dependent. Now, we know that it can be overwhelmed. So what we wanted to do next was ask the question, well, what is the dose threshold? At what point are these cells and the liver going to be overwhelmed? And so we answered this question using an in vitro experiment, right, where we increased the dose of nanoparticles that we gave to these in vitro macrophages, and we just measured how many particles per cell were in each of these cells. And so we found that as you increase the dose, there was an increase in the particles that accumulated in the cells, of course, but it flatlined, right? So after a course of 24 hours, what we found was that the maximum amount that a macrophage could take up in vitro was 100,000 nanoparticles per cell. And so we started to do some estimations, right? At 100,000 nanoparticles per cell, and also at 10 million cover cells per mouse liver, if you multiply these two numbers together, what you find is that should be about a 1 trillion nanoparticle per liver uptake limit over the course of 24 hours. That's exactly what we saw over here, right? When we looked at the liver, around this area of 1 trillion, we started to see this inflection point that showed us that the liver was beginning to be overwhelmed. And so I want to spend a little bit of time on the mechanism now, right? What is being overwhelmed? And we had 2 hypotheses. It was either the uptake rate that was being overwhelmed, so a kinetics point of view, or it was the uptake capacity that it was being overwhelmed. And for the uptake rate, you can imagine we are giving so many nanoparticles and they're zooming by the liver and the cover cells so quickly that the cover cells don't have enough time to grab them all out of the circulation, right? And on the other hand, the other hypothesis was, well, maybe they just, the cover cells have eaten so much and they're so full that they just can't eat anymore. And that would be overwhelming the capacity. And these two, we wanted to figure out exactly what was going on. And so to assess the rate, what we had to do was we had to do intravital microscopy, right? That gave us fine temporal resolution. And to show you what the intravital microscopy looks like, it is live animal imaging. We have a live mouse that is under anesthesia on a microscope. And essentially what we've got here is we've got an anesthesia machine and it's hooked up to the microscope. We have a mouse that's asleep and that's opened up, and the lens is pressed directly against the organs, and it gives us a live feed of what the cells are doing in real time. And you can see here, we've got a couple of cells in blue, we've got nanoparticles in red, we've got nanoparticles in green, and that really gives us an idea on a second-by-second basis of what's going on inside of the organs. And so to quickly give you the results of what we had shown, To do this experiment, we wanted to compare the uptake rates of two doses, right? One dose that was below the uptake dose threshold, and one dose that was above the dose threshold, the high dose. So we call them low dose and high dose here. And essentially what we had done was we had labeled these nanoparticles with fluorophores, right? And we imaged in that channel of the fluorophores. And what you'll appreciate here is that over the course of 25 minutes, this cover cell that's shown in blue has taken up quite a few of these Psi3 labeled gold nanoparticles, right? And then what we wanted to do then was we wanted to compare this to the high dose situation. In the high dose situation, we had supplemented these Psi3 nanoparticles with an excess of Psi5 nanoparticles. So they're labeled with a different color. So we wouldn't be able to see them in the Psi3 channel. And so what we showed here is that over the course of 25 minutes, there was a very minuscule amount of uptake in these cover cells, which suggested that there was some sort of competitive inhibition going on, right? Because these cells were taking up these other nanoparticles, they could take up these Psi3 nanoparticles much less, right? And then at the end of this, there was the question of, what if the nanoparticles had saturated their capacity within the first 30 seconds, right? Because the dose was so high. And so to answer that question, we looked at the nanoparticle uptake in the high dose channel. And what we found was that over the course of 25 minutes and even up to an hour, there was continuous uptake of nanoparticles, right? Suggesting that the capacity was never actually overwhelmed. And we just show this in graph form, the low dose and the high dose. This is the uptake over the time course of minutes. And what we see here is that we have a significant difference in the uptake rate between the low dose and the high dose. And if we image in the high dose channel, we find that there's continuous uptake of nanoparticles over the course of 60 minutes. And so we conclude that the uptake rates are overwhelmed, but the capacity is not overwhelmed. And not to kick a dead horse or anything, but we really wanted to verify using a different method if the capacity was overwhelmed, right? And what we found using transmission electron microscopy, we wanted to estimate the volume that these nanoparticles were taking up, right? So 15 minutes after the injection of nanoparticles, at this point, things had already been overwhelmed and the uptake was slowed. We wanted to say, do the nanoparticles take a big proportion of the cover cell, right? So we're back in the liver, this is a cover cell. And you can see that the nanoparticles here represent maybe about 5% of the volume of this cover cell, right? And I'll tell you that this is nowhere near the maximum capacity of a cover cell, right? So when we look in vitro and we give as many nanoparticles as we can, these cells can take up ridiculous amounts, right? So this is a macrophage in vitro. And what you'll appreciate here is all of these black dots, these nanoparticles, they've taken up 60 to 70% of the capacity of this macrophage, right? And so there is plenty of room left to take it up. So cover cell capacity is not overwhelmed. And I spent a long time on this mechanism, and it's a very subtle but very, very important point right here, right? And I will promise that I will come back to this point nearer to the end of the presentation to explain why it's so important. But for now, we want to hear about the tumor, right? We've heard a lot about the liver and how this works, but how does dose affect the tumor? So what we did was we measured the delivery inside the tumor, and again, we see the exact same thing, right? We see that there is some dose threshold to get increased delivery to the tumor, right? And we see that as you increase the dose, we start to get this increase in the delivery to the tumor. And at the highest doses that we gave, we saw a delivery efficiency of 12% of the injected dose inside of a tumor, which is orders of magnitude better than the current median, right? And this shape was mirrored in the liver. The liver went down as we increased the dose. And we can get a bit of a better understanding of this if we take these two these points and we divide them by each other to create a tumor to liver ratio, kind of like an index, right? The tumor goes up, the liver goes down. And what we see is that it's quite clear now, right? We start to see a log linear relationship where at low doses, you get very minimal accumulation of the tumor. Most of it is in the liver. And somewhere around this trillion threshold, we start to see this increase. And importantly, I want to point out this point right here. At the highest dose that we gave, the tumor was actually able to take more nanoparticles than the liver on a gram per gram comparison than the liver, suggesting that the tumor has this capacity to take up more nanoparticles than the liver, which to us at that point was a wild idea, right? So we wanted to make sure next that our threshold and our theory was generalizable, right? we wanted to ask, well, is it a function of just our nanoparticles? Is it just a function of the tumor model that we were using? And what we found was that no matter what tumor model we threw at it, we saw the exact same effects, right? So we ran some xenogenetic models with human breast cancers in immunocompromised mice, also human brain cancers, also mouse spontaneous cancers in immunocompetent mice, and also just validating the 4T1 model in a different immunocompromised mouse. And what we saw, no matter what we did, was that if we increased the dose past the threshold, we got the increase in delivery to the tumor in all of these, and we saw the decrease in accumulation in the liver, right? Which suggested to us that this was biology independent. This was just something that was a phenomenon that was generalizable across biology. And when we checked different nanoparticles types, we saw that the exact same thing. As you boost the dose past the threshold, you get this increase in delivery of that tumor to liver index, right, of silica and liposomes. And so then, we asked the question, okay, so the tumor seems to accumulate a lot more. We want to next ask the question, well, what happens to the nanoparticles that get in the tumor, right? And we hypothesized that, if you can get more nanoparticles into the tumor, then these nanoparticles would, just by pure diffusion, would be able to penetrate deeper into the tissue, right? And this is really important because one of the limitations of cancer chemotherapy, whether it's a nanoparticle or it's a small molecule, is that they're limited by their diffusion distance, right? They just can't get deep enough to hit all the cells of the tumor. So we hypothesized that, if you give a higher dose, you should be able to get deeper into the tumor. And that's exactly what we saw, right? So here we did 3D microscopy to visualize a volume of the tumor. And we have blood vessels in red, and we have nanoparticles in green. And what you'll appreciate here is that at the higher doses, there's a much more diffuse distribution of our nanoparticles away from the blood vessels, right? And when we quantify this, what we find is that the distance from the vessel that these nanoparticles get to, if we look at a higher dose, the nanoparticles penetrate further than if we were to give a small dose, right? So this verified our hypothesis that the penetration would be increased. And, if we're going to get further into the tumors, then we should be able to access more of the cells, right? And that's exactly the case, right? So when we gave a lower dose of nanoparticles, here we have cell nuclei in green and cell nuclei in blue and nanoparticles in green. And what you'll see here is that the lower dose, we have a few speckles of green here and there. But really, when we increase the dose, we see a rather homogeneous distribution of our nanoparticles throughout the cells of the tumor, right? And when we quantify this on flow cytometry, what we find is that at the highest dose, we had 93% of all cells in the tumor had taken up at least one of the nanoparticles, right? Suggesting that we could access almost all of the cells in the tumor by increasing the dose. Now, when we ask the question, well, where exactly are these nanoparticles inside the tumor? When we look at transmission electron microscopy, you can appreciate that this is the outline of a cell. And the majority of the nanoparticles are inside these endosomes of the cell, right? And a minority of them are in the extracellular matrix out here, right? So the majority of nanoparticles are inside the cell. And indeed, when we look again at flow cytometry, the more you give, the more uptake per cell that we saw. Now, we were worried that maybe this is an artifact of our lab, right? Maybe there was something with our measurements, maybe there was something with our particles or our model, and that we were biased towards these measurements. And so we wanted to confirm, what about other studies? Has this been shown before, right? There are so many published studies of nanoparticles out there. We wanted to see, well, maybe in these other studies, if we see this effect, then this should be a widely applicable effect, right? And indeed, we looked back at the meta-analysis that our lab did back in 2016. There were 117 papers associated with that meta-analysis that represented the field for 10 years. And what we did was we went back and we recalculated every single dose that was reported in those papers. And we turned it into the numbers of nanoparticles, right? And what we saw was that indeed this threshold and this dosing exists throughout literature, right? And so as you increase the dose, what we saw was this inflection point over here that roughly happened around 1 trillion, right? And when we divided the studies in half, it turned out that the median was about 1 trillion nanoparticles, right? And we divided them in half and we compared them to each other. And you can appreciate that nanoparticle studies that gave below that dose threshold, there was less tumor delivery than nanoparticle studies that had given doses above that threshold. And then we wanted to ask, well, is this robust, right? Is there some confounding variable in nanoparticle design where the higher doses were just maybe they were smaller sizes, or maybe they were active targeting, or maybe there was some material, right? And so we started to break down into these subcategories. And what we found was that the dose threshold is extremely robust no matter how we decided to do this, right? So we broke it down into different types of nanoparticles, whether they're organic or inorganic, and you'll appreciate that the pattern remains. And we get the same kind of comparison when we look at their individual classifications. So no matter what, if you gave a higher dose above that threshold, we observed that the studies showed increased tumor delivery, right? We saw this for passive versus active targeting also, right? So if you if you added a targeting ligand that specifically recognized cancer cells, you get the same increase no matter if it had that ligand or if it didn't have it, okay, right? And then finally, we also looked at size, which is an important nanoparticle design parameter, and we see the exact same thing, whether they're small particles or big particles, as long as you gave more, there were more that entered the tumor, right? And so we said, okay, well, among all of these parameters, which one seems to be the most important, right? And when we ran a multivariable analysis to look at the contributions to delivery efficiency of these different particles, what we found was that dose was the overall, unwaveringly, the most significant design parameter that affected tumor delivery, right? So the interesting thing is, in those 117 papers that we looked at, not a single one of those papers rationalized their choice of dose, and only one of those papers used more than one dose, right? And so this suggests to us that this dosing parameter is so far, it's very significant, but so far it's been unrealized, right? And so we've done a lot of delivery efficiency, measured how the nanoparticles can get into tumors, and we developed the principle. The next step was to ask, can this be translated, right? Can we invent a solution that improves the treatment of cancers, right? I know this was a little bit of a tricky experiment to do at the beginning, right? It was tricky because you can imagine if we were to give a nanoparticle that was loaded with a drug, then what would happen was if we increase the dose, then we would also be increasing the dose of the drug, and the tumor would shrink a little bit more, right? And so that would be obvious, and it wouldn't really prove our point that the nanoparticle dose is the thing that really matters. And so what we did was we came up with a solution to isolate the nanoparticle dose by number in our efficacy studies. And so we chose calyx as our model therapeutic nanoparticle. So this is a clinically approved nanoparticle that's used to treat cancers in human patients right now. It's a liposome, so it's made of lipids, and it's loaded with doxorubicin on the inside, which is an anti-cancer drug, right? What we did to make sure that nanoparticle number was important was we looked at delivery enhancers. We created these nanoparticles called delivery enhancers, and they were liposomes, and they were liposomes that were not loaded with any chemotherapies, right? So they were just empty nanoparticles, and their main goal was to be able to boost that dose artificially and distract the liver. And so we had 4 conditions that we compared. We compared animals that got a sugar control, animals that got just delivery enhancers, animals that got the calyx only, and animals that received a co-injection of both the delivery enhancers and the calyx. And what we found was that when we compare the delivery enhancers to a sugar control, there was no difference in the growth of the tumor over time, right? And we're looking at these gray lines over here, suggesting that the delivery enhancers had no therapeutic efficacy on their own, which makes sense because they're not loaded with any sort of drugs, right? Then when we injected calyx, what we found was that we got this reduction in the tumor volume, which makes sense. They're loaded with a little bit of drug. But what was really wild was that when you just added the delivery enhancers with the calyx, we saw an additional 60% reduction in the tumor volume, which suggested that really, if we are to increase the dose artificially with these delivery enhancers, it would allow us to enhance the anti-cancer therapy of currently existing nanoparticles, right? Boosting the dose improves the efficacy. And we saw a small increase in the survival, extended the lives of these mice by about 30%. And the big question is, well, in chemotherapy, if you're going to reduce the tumor size, you're typically introducing some sort of toxicity, right? And so we tried to look for the toxicity, and what we found was no matter how we studied this, the delivery enhancers were not toxic. right? So we looked at our mice and we studied their blood. We looked at the distribution of cells within the blood. We looked at the electrolytes. We looked at lipid profiles, and we also looked at the liver function tests. And among all of these, the values were normal, suggesting there was no toxicity to the blood. We also looked at the liver histology to see if there were some sort of pathological changes, and we found that in the liver, everything was normal. And we also looked at the heart because doxorubicin is classically known to cause a lot of cardiotoxicity. And we asked, is this happening here? And we found that the heart was also normal, right? And we looked at all of these tissues at 2 days acutely, we looked at it subacutely, and we also looked at them chronically. And what we found was that there was no signs of any sort of toxicity at any of these time points, right? Which suggests to us that the delivery enhancers at the dose that we used, they were not toxic. Now, I had promised you that I would tell you why the mechanism of clearance was important, why uptake rate versus uptake capacity was an important consideration to make. And here's where I will tell you why, right? So there's a technique that exists called the RAS blockade. And the RAS stands for reticuloendothelial system. And it's essentially the cover cells of the liver plus a couple of other clearance cells in other organs. And what we wanted to do was we wanted to ask the question, how does this compare to the RAS blockade technique, right? How does our dose principle line up with it in a comparison? And the RAS blockade technique, what it does is it injects a bunch of empty nanoparticles first, and it waits for the cover cells to take them up, and they get saturated in terms of their capacity to get full. And then after a couple of hours after that, then you inject the therapeutic nanoparticles. And after you do that, the therapeutic nanoparticles are supposed to skip and avoid the cover cell clearance and supposed to give more delivery to the tumor. And so we did these injections, right? We said, okay, well, ours says that it's an uptake rate, so we should maximize the concentration, and theirs is a uptake capacity, and so you should maximize some sort of waiting time between the two injections. And what we found was that no matter what we did, the co-injection proved to be better than the sequential injections of the RAS blockade. Right. So we looked at the blood 24 hours after injection. And what we found was that there was more nanoparticles left in the blood if we gave a co-injection versus if we had a short delay or a short delay of 15 minutes or a long delay of 24 hours. Right. And as you increase that delay time, the amount that's left in the blood decreases. And same thing in the tumor. Right. As we increase the delay time, the amount of tumor delivery also decreased. And this translated to a small but statistically significant difference in their tumor sizes. So when you gave a co-injection of your nanoparticles, you had up to 30% reduction in the tumor sizes, as opposed to if you were to delay these two injections, right? And it translated to a little bit of survival time. And this makes sense, right? Because if the mechanism of overwhelming the liver is truly because of the uptake rate, then you would want to maximize the amount that's in the blood circulating at any given time, right? You can imagine that if we were to give sequential injections, then that time delay that is in between these two gives time to allow the cover cells to start taking nanoparticles out of the blood, so that by the time you inject the therapeutic nanoparticles, those therapeutic nanoparticles combined with the empty ones, it's already at a lower effective concentration, right? And so, so this is why the uptake rate is so important, right? Now, we've done a lot of studies. I wanted to lead you through a little bit of our thoughts and our discussion about humans. What are the human implications of this, and how might this be translated to the clinic? So we did a lot of mouse studies, right? And we want to ask questions about it. The thing is, the mouse are much smaller than humans, right? The mouse liver weighs approximately 1 gram, and the human liver weighs approximately 1,500 times larger than that, right? So if we make some quick assumptions about the human liver, assuming that the cover cell density and the cover cell activity is roughly the same as a mouse, then we can make an extrapolation based on mass. And so this 1 trillion threshold that we have in mice corresponds to 1.5 quadrillion in humans, right? And so what does this mean? Is this just some random number that we're thinking about? So we tried to look at as much clinical evidence that we could that was already published. And we said, okay, clinical nanoparticles should be given above 1.5 quadrillion. What are they given at today? And so you look and you see that some of the successful nanoparticles that are used in humans today, they're given at doses above this 1.5 quadrillion cutoff, right? And interestingly, if you look at some of the high-profile nanoparticles that missed their clinical endpoints in their phase 2 clinical trials, they're given at doses that are below this 1.5 quadrillion threshold, which, this could be a coincidence, right? But for us, it pointed out that question that, what if those nanoparticles had been given at just slightly higher numbered doses, right? Maybe they had been combined with empty nanoparticles, maybe they had been loaded with less drug per nanoparticle. But if they had been given at doses above 1.5 quadrillion, what would have happened? We don't know. So I think a clinical trial will have to come out of this, and we can assess the validity of this threshold in humans, right? And so, at the end of all of these experiments, there are a lot of questions left to explore, right? And I just wanted to share some of those with you today. And the first one that I'm really curious about is the mechanism of how the cover cells are overwhelmed, right? When we were looking at this, what we saw was that these nanoparticles inside cover cells at short time points, they always lined the perimeter of these endosomes, right? And we rarely saw nanoparticles inside the lumen, which suggested to us then that, these nanoparticles have to stick onto the membrane in order for the nanoparticles to be internalized, right? So then the question becomes, right, how do nanoparticles get inside? What are these ligands that the nanoparticles are binding to? There should be some sort of receptor-ligand interaction. And so the main questions that I would have then are, what are the receptors, number one, and number two, how many receptors are binding per nanoparticle, right? And we have a great new grad student in our lab that is going to be exploring this, and hopefully we'll figure out an answer soon. And I'm really excited to see the results, right? The next thing, the mechanism of tumor delivery. This is an age-old question in the field of nanoparticles, and the dogma that had governed nanoparticle tumor delivery for so long. The idea that was passed around in the 80s and 90s, that the nanoparticle tumor vessels are immature and leaky, and they had gaps. I think that is started to be in question a little bit now, right? And what ends up happening now is we're not sure. We don't think it's a passive delivery mechanism. Maybe it's an active delivery mechanism. And so I'm sure a lot of us have thought about this after injecting our nanoparticles and waiting for that 24-hour time to work out. But we're wondering how exactly our packages get there, right? And this our lab, led by Dr. Sindwani and Dr. Syed, earlier this year, we had brought that idea of the passive accumulation into question. We don't think it's a passive accumulation mechanism. It's something active, and this will be figured out. And we've got grad students that are working on it now, and hopefully we'll, again, be able to figure out what's going on, right? Next, I want to talk about gene delivery. Gene delivery is so hot right now, super exciting. We've got Alnylam that had just had their approvals on Patcho with their lipid nanoparticles. And we've got a lot of companies that are looking for how viruses can also carry genes into different tissues, right? And so I'm bringing up this clinical study of a phase 2 clinical trial. What they essentially did was they were replacing one of the enzymes that was missing in a couple of patients and replacing them in the liver. And I bring up the study because I found the doses to be extremely interesting, right? If you look here, they broke down their patients into 3 different doses: the low dose, an intermediate dose, and a high dose. And they were giving these patients viral particles loaded with genes. And what we see here is that the endpoint that they measured, the end result that they measured, the factor VIII activity, in the low dose and the intermediate dose were low to non-detectable, right? So what dose did they use in the low and intermediate? It turns out that for the low dose, they used 0.4 quadrillion viral particles, and the intermediate dose, they used 1.4 quadrillion nanoparticles, right? And so this kind of suggested to us that if you were below that dose threshold, it may also apply to viral particles and gene delivery. And their high dose was dosed at 4.2 quadrillion viral nanoparticles, and they ended up with robust responses that were still detectable a year after their therapy, right? So now they're running their clinical trial, their phase 3 clinical trial in many more patients, giving an average dose of about 2.8 quadrillion viral particles per patient. And we believe that because it's above that threshold, we think that trial will be successful and things will work, right? And finally, I want to talk about the toxicity, right? We didn't find any signs of toxicity, which then suggested to us that potentially we could increase the dose, right? We can increase the dose and we can boost the delivery a little bit more. So what I want to do is it can be an interesting experiment to create the high dose that can increase that delivery until we start to see signs of toxicity, and then we would know the limits of this technique. And so that brings us to nearly to the end of this presentation. I just want to give some conclusions and some take-home messages. But before we do that, I want to give a little bit of advertising for this paper. We published our paper last week and a half ago, and we're really excited to share it with everyone. This was a process that took us 4 or 5 years with many different people, and we've been working on this, and we've learned so much about how nanoparticles behave in the body through this process. And today I presented just a minor fraction of this paper. Right? We have 41 different supplementary figure panels, and each of those taught us a new fact about how nanoparticles behave. So we hope that you'll give it a read and that you'll reach out to us with any questions that you have. We really invite discussions, right? So in conclusion, we have that question, right? How do we overcome the liver, right? And this set the stage for the majority of my thesis. And I went about answering this question with 3 different methods, right? 3 different themes. First, we wanted to understand the system. And that showed us that the liver macrophages are intravascular, right? They are within the blood. They have preferential access to the nanoparticles upon injection. And then so we used that to develop our principle to show that, that preferential accumulation in liver macrophages can be overwhelmed. That dose really determines tumor delivery, but that we have to breach a threshold dose to be able to get to that tumor. And this allowed us to invent a solution, right? We were able to use empty nanoparticles to supplement and enhance anti-cancer therapy just by boosting the dose. And so with that, that's the end of the scientific content of this presentation. But today I have the honor of presenting the work that we had done. But really, this was the efforts of a large group of people over 4 or 5 years. And I really just want to tell you, I can't claim all the credit for this work. So I want to acknowledge them today, and I want to tell you their stories. And hopefully you'll stick with me. This is going to take a little bit longer, but I hope that you'll find the stories to be amusing and fun. So when I go back and I think about my favorite memory in the lab, right? It's never a single memory. It is something that occurs over, that seems to be reoccurring. And I'll just paint a picture for you for what that looks like now. So I'm in the lab, and I'm wearing a lab coat, right? And in front of me, I'm sitting in front of the chemical fume hood, and there are hundreds of samples in front of me. I look to my immediate left, and this man is sitting there. This is Yinan Zhang, right? And he's holding a bunch of samples too. And I look to his immediate left, and I see Wilson, right? He's got tons and tons of samples. And essentially what's going on is that it's ICP-MS day, right? We're looking at biodistribution of organs. It's extremely monotonous manual labor, right? We're literally mixing hundreds of samples and we're washing tubes and we're pouring water and acid around. And the work itself is terribly dull, terribly boring. But I always looked forward to these days because they were so much fun, right? We, the 3 of us, we would laugh, we would joke around, we would catch up on with each other, and there would be Jay Chow playing in the background on the speakers on full blast. And for those of you that don't know, Jay Chow is an Asian slow jam singer, and so we would be singing along, and it was just generally a really great time, right? And I really appreciated the fact that we could turn a monotonous task into something that was exciting and something that we could look forward to. And so Wilson and Yinan have been with me since the very beginning. They've seen the inception of the dose idea, and they're like my brothers in the lab at this point, right? And they've taught me so much, right? I've run lots of leadership positions with Wilson. We've run a conference together, and he's taught me lots about leadership and lots about connecting the dots between different groups that do nanomedicine in different papers and different labs. And Yinan has taught me lots about a positive attitude while working. He's always happy, always cheerful, always energetic. He's taught me about persistence and the value of that attitude, right? And so this is our last picture together that we took before Dr. Zhang headed off to California, and he's going to be studying vaccines, right? Dr. Poon is going to be going off to California as well to study genomics, and I'm absolutely confident that they're going to make fantastic discoveries, and I hope everybody will follow their pathway because surely it will be exciting. And I want to tell you about some of the other people right now. This is Zachary Lin. Zachary Lin and I learned intravital microscopy together over the course of maybe about a year and a half. And it essentially involved sitting in a dark room for hours, turning a focus knob every 3 seconds, right? Really, really extremely difficult work, but it was made so much better with Zach's presence. And he's now the expert in intravital imaging, and he's also the expert in making our delivery enhancers, right? And he's sitting here. This is when we gathered at my place to just kind of hang out and sing karaoke. But Zach doesn't like music that was made after the year 1850, right? And so you can imagine we're singing our pop songs from the 2010s. Zach wasn't very fond of that. So what he ended up doing was he put on noise canceling headphones and he just jammed to his own thing and left us alone. It's a really funny guy. This is Benjamin Kingston. This is 2 bends in front of a gentle bend chairlift at one of the ski mountains. And Ben Kingston is our expert in 3D imaging and machine learning, right? And so the 3D images that I had shown you earlier, Ben Kingston had collected them on the last day before the COVID lockdown. We were sitting in the hospital in this lab and wondering, are we going to be kicked out? Will there be COVID floating around? And we weren't really sure. But that really just shows you what an amazing teammate he is and that dedication that he has, right? So glad to have him on our team. This is Dr. Anthony Tavares, and I think that Dr. Tavares represents the epitome of a rigorous scientist who knows everything down to the very first principles, right? He has taught me everything I know about physical chemistry, and he led our charge in getting the silica nanoparticles to work and to conjugate them and to measure their biodistribution. And the meme here is that we would be working in the lab, anybody, and we'd be concentrated and very productive, and you would just kind of hear out of the corner of your ear, "Yo, Ben," and you would be broken from your concentration. You'd look up, and it would just be Anthony doing this, right? And he would laugh it off, and then he would walk away. And so he was an incredible presence in the lab, made everything so fun, invented so many phrases, and he would hit you with these 2 points of voice at any given time, right? Over here is Johnny. Johnny is our protein corona expert, right? He, because of him, we figured out the differences in the protein corona between the liposomes and the golden nanoparticles. And he was instrumental in figuring out why that was the case. And we were able to then conclude why these nanoparticles seem to go to different cells, right? A little bit more research to be done, but we got a little bit of hints there. Now, this is a picture of Johnny in a tent. We'd gone camping, and he's making this face. He's making this face because the night before, Ben Kingston had made us some bean tacos. And Johnny and I were tent mates, and the bean tacos didn't sit so well in my stomach, right? And we'll just leave it at that. Johnny was pretty upset with me for a while after that. So I apologize, Johnny, but you know what? I would do it again. Right? I want to introduce you to Syed and Presley over here. Syed pretty much invented the visualization of the nanoparticles inside of a 3D tumor, along with Trace and Wani. So we're extremely grateful for him for introducing this technique in our lab, and he's also a bioconjugate whiz. Presley has taken over his role. She's going to be looking at more of the distribution of nanoparticles in tumors in 3D. Along with Syed and Jess, Presley has created a new technique to be able to visualize liposomes inside of 3D tumors, which is a really amazing feat because, during the processing, for clarity, you actually get rid of all the lipids, right? So to be able to see liposomes is really neat. And over here, we have just, this was last year, we had just run the CIBC Run for the Cure for the Canadian Cancer Society. Where as a lab, we ran this, and from very many generous donors as a lab, we had raised over $2,700. So it was a really great experience, really great run for a fantastic organization. And, we're running it again this year. So if you have a couple dollars, please send it our way, send it to the Canadian Cancer Society. It's for a really great cause. And I want to introduce you to Julian Couture-Senecal over here. We met Julian 4 years ago, he was in his first year of undergrad, and he joined our lab as a summer student, and he helped us optimize a lot of the in vitro experiments. And he had a fantastic energy, brought so much vitality, and he was really able to grasp those big ideas very easily, right? Every time he read a paper or just understanding the conversations, high-level conversations that we had in the lab. And he's been really supportive. This was at the end of one of my presentations, where he made a poster and wore it for the presentation. So he's starting his PhD now in BME at U of T. So we look forward to seeing where he goes. And of course, Warren Chan, the big boss. I have so many words to say for Warren. And I'll share a story with Warren as well, right? So we won't talk about the undergrad stuff. We'll just keep it to this. So when I first met Warren in his lab, and I emailed him to ask for a 20-minute meeting to see what his lab was doing and to see if I would be a good fit. That 20-minute meeting turned into a 2-hour conversation where Warren asked me questions about why I was interested in science, what did I want to do with the PhD, and what did I want to do with my career, right? And for a student that wasn't in his lab, he had spent 2 hours talking to me, and that really exemplifies the kind of mentor that Warren is, right? And these conversations have happened throughout grad school now, where we've met and we've just chatted for hours on end, right? And so, over the years, Warren's been the best mentor that I've had, and he's taught me so much about focus, about doing rigorous science, and about communicating, speaking, and writing. And after 5 years, it's surprising, I think, that when I think about it, there is still so much more that I have to learn from Warren, right? So hopefully in the next couple of years, I'll continue getting more lessons from Warren, right? And really, a big thanks to Warren for essentially building our lab, right? So this is our most recent lab picture. This is the Chan lab. And this is our green themed photo, in case you were not sure. And really, Chan Lab, I really want to thank you guys for making this such a fantastic time. There's so many personalities, and everybody really adds a little bit of dynamic and vibrance to the environment. And I wake up every day and I'm excited to go to the lab because I'm not sure what the day's joke is going to be, or if there's been a paper that was published recently, we're going to spend 3 hours debating all of the experiments of the paper, or if we're just going for coffee. Right. So thank you guys for making such a fun time to work with over the years. And especially thanks to Dr. Fai Song over here, who is our lab manager and who has made everything go so smoothly over the last couple of years to make sure that our lab is always running. Right. I want to thank our collaborators, Dr. Gong Zhang, his lab members, Juan Chen and Michael Valek. And essentially, they were instrumental in helping us figure out the biodistribution of liposomes. They lent us their radio labeling techniques so that we could test the dose threshold principle with liposomes. I want to thank Milan and I want to thank Vivian for running so much of our histopathology and making sure that we knew the toxicity and that we knew where our nanoparticles were going. I don't have a picture of Kyle Duffin, but he's also here too, who helped with a lot of our biochemistry. Here I want to thank Kim Soy and Sonia McParland. Kim and Sonia led the charge in the nanomaterial clearance from the liver mechanism. When I first joined the lab, Kim had taught me a lot about how nanoparticles are cleared from the body. She taught me a lot about rigorous science, and she taught me how to make nanoparticles for the very first time, right? And over the years, she's offered many wise words of advice. And Dr. McParland has been our residence immunology expert. And so over the years, she's provided excellent guidance on that. And Stefan Wilhelm, he led the initial charge for the 2016 meta-analysis. And so thank him for setting up the foundation of my project in the field. Thanks for also the consultations he's provided over the years, right, on how the dose concept also applies to all of the papers that he analyzed. And this is a paper that Wilson Poon made for me. He's an excellent artist at digital communication, and he made this to advertise the defense. And essentially, this is Harry Potter themed. We've got our lab over here. We've got Hedwig over here, but instead of Hedwig, it's a mouse with wings. And this is our streetcar that we use in Toronto. And I showed this poster really because I want to thank my committee members, especially to Dr. Gong Zhang and Dr. Sergio Brinstein, who have provided so much insight into material science and cell biology over the years that have really guided the course of the research project. This is Dr. Margaret Cheng, the internal, external for my exam today. And it's really cool because it's kind of come all the way around because she was my qualifying exam, internal, external. And I remember at the end of my qualifying exam, I had written 4 aims, but I only presented 3. And Dr. Cheng picked up on this and she said, "Oh, well, I noticed that you didn't present aim 4. Do you think you'll be doing it?" And so today, at the end of the presentation, I've shown you that I did not present aim 4, and also did not present aims 1, 2, and 3. I hope you're not disappointed, but we found such a cool effect while we were looking for those aims, and that we latched on and we couldn't let it go. So I hope that you're not disappointed and that the work today that was presented is still interesting. And I want to thank Dr. Yoon Yao, who joins us from Purdue University today. I had met Dr. Yoon Yao at the Gordon conference last year, and we had lots of pleasant discussions on nanoparticles and how they worked. And at that time, I thought that she was extremely nice. I was really excited to have her as my external examiner. But recently she's been hinting that she's going to make me cry after the end of grilling me for this defense. And so I'm a little bit wary, but I hope that, she's just kidding. And there are a couple of people that I have to thank. I'm almost done, I promise, that I have to thank that were outside of the lab, not directly involved with the research, but had made a big impact, right? And so first, I want to thank this group of people. These are my friends who eat spam, and they will understand what that means. Hi, guys. And essentially, we had started med school together, and I left to do a PhD, and they continued on their medical education. And they are all senior residents now, or some of them are staff doctors. They've been super supportive and very patient with me over the years as they waited for me to finish my PhD and join them on the wards again. And I'm really excited to tell you guys that, med school starts next week. So I'll be seeing you on the wards as early as then. So see you guys around. This is my MD PhD cohort, which I started with back in 2013. And it was really great to be in a group of people that whose personalities were so tightly knit, and everybody shared the same career aspirations to become a physician scientist, right? And we're coming to the end of our journey. We're almost done, and it's really exciting to see where everybody ends up. This is my lovely girlfriend, Cynthia. This is when we had traveled to Egypt. Cynthia has been with this PhD process pretty much from the very beginning. And she's seen the ups and downs. In research, not everything works all the time. And so she's always been there for me when I've been stressed out. And she's always been the first to celebrate with me when things go well. So thank you, Cynthia, for that and for teaching me essentially how to be kind, how to be patient, and generally how to be a good person. And for teaching me the joys of traveling. This is Egypt that we had traveled to, the last place we had traveled to together. Really great time. And really, Cynthia's been the first person who has taught me truly that it is okay to not work sometimes. And lastly, these are my parents. This is Jianfei Ouyang, and this is Shuyan, my mom and my dad. And so they have spent 30 years raising me, and they have dedicated their lives to me in making sure that I would succeed. Right? They've removed all barriers that I've had so that I could focus on my education and that I could succeed in my dreams to become a clinician scientist. Right? They've stimulated my curiosity. They've established my personality. They built my character, my grit, and built that foundation of who I am today, so that I could be who I wanted to be tomorrow. Right. So I really want to thank my parents, Baba, Xie for doing that, for being there for me. And really, at the end of all this, it's been a wild 5 years. And I reflect and I think back, and I think we made a lot of really cool scientific discoveries that we'll remember over the years. And I think most importantly, though, what I'll really remember over the years, the memories that are really precious, are the times that we had all spent together. And so, truly, deeply from the bottom of my heart, I want to thank everyone who's been part of this journey for the last 5 years. It has been a really, really wonderful time, some of the best 5 years of my life. So thank you so much for being part of my journey. And to the friends and family who have attended the PhD Defense today, thank you for taking the time out of your day and your busy schedules to listen to me ramble for an hour. And with that, I'll conclude the presentation. Thanks so much.